I want to cross over to Dan Fornika. So Dan is from British Columbia CDC over in Vancouver. He seems to have made a start with HPV and then moved into bacterial genomes. He identifies himself as a molecular biologist and bioinformatician, so that's a genomics specialist. And his recent software, Mentalist, is excellent for rapid CGM listy typing from short reads. And today he's going to be talking about the Irida platform and developing pipelines for it. So let's cross over to you, Dan. Where are you?
Yep, I'm here. Can you hear me?
Yeah, we can hear you. Yep.
Okay, I'm just trying to figure out which screen to share here. First, I'll just briefly correct you, Nabil. Mentalist is a great piece of software, but I'm not the primary author of the software itself. There's a fellow named Pedro, who is a postdoc who wrote most of the code and the algorithm, and I played a supporting role in helping to maintain the software and help the GitHub repo to stay in good shape and make releases and that sort of thing.
My apologies.
But Mentalist is definitely a great, fast way of doing core genome MLSD. All right, so, and as a nice segue, it's also included in the Irida platform, which is the subject of my talk today. So today I'm going to do a brief intro on Irida pipeline plugin development. So let's, okay, here we are. Just as a quick outline, there are basically three main topics I'm going to talk about today. A quick intro to what Irida is and why you might want to use it. We're going to talk about how to find and install Irida pipeline plugins that already exist. There's a growing suite of existing pipeline plugins out there that I hope some of you might be interested in adding to that. And there's a single place where you can look to find some of the existing plugins that are available to you right now. And then we're going to talk a bit about how you can develop your own Irida pipeline plugins and how to set up your own development environment to make it convenient to test and develop and break and fix your plugins on your own system without having to interfere with any other existing Irida system that's out there. So first, what is Irida? Irida stands for Integrated Rapid Infectious Disease Analysis. It's an open source end-to-end platform for public health genomics. I've got a couple links there. I hope I'll be able to share my slides so that people can click on all these links, because I've included a few of them. But the first one is simple, irida.ca. That'll give you lots of information on the overall platform and some of its design goals. And there's also a GitHub site because it is all open source. You can see all the source code, and if you have any requests or additions or bug fixes, or those sorts of things, then it's a very actively maintained project. So the GitHub site is a great resource as well. So we have a broad overview of some of the Irida features. So we'll start with data management. The main sort of entities that Irida deals with are projects and samples. You can have a huge number of projects in your Irida server, and each project can have a number of samples. This is really useful for sort of long-running investigations, surveillance projects, where you might be accumulating samples over the course of months or years, and you want a sort of persistent place to consistently store your samples, and you always know where to go. You can just open up a web browser and see your list of samples. It's also possible to share samples from one project to another. And along with that comes some user accounts, so you can add specific people to the system and give them specific access controls. There are, of course, analysis pipelines. Irida comes with a curated core set of pre-installed analysis pipelines. And there are additional pipeline plugins available. That's what we're going to be talking about later on. Behind the scenes, the pipeline execution environment is Galaxy. So if you have any experience with Galaxy, the Galaxy platform, then you're already halfway there. If you've ever developed a Galaxy pipeline, then you're very close to already having an Irida pipeline plugin. Irida also gives a lot of nice features around data integration. So it provides one feature that is just critically useful, and I think at this point maybe a bit underappreciated, is there's a very consistent, nice REST API. So all of the analysis results and sample metadata that's stored in Irida is accessible programmatically. So you can write a Python script or something that will pull data from the Irida REST API and manipulate it or further analyze it. And along with that, there's a integrated metadata line list. So for each project, you have a line list, a spreadsheet where you can store both some of the analysis result details and also other custom pieces of metadata, like the sample type or collection date or those sorts of things. It also has some nice visualization components. There's an interactive phylogenetic tree viewer, and you're also able to overlay some of the metadata from your metadata line list alongside the tree. So that can be really useful. So I'll just give a quick, now that I've explained everything, I'll just give a quick tour to show off what this all looks like. So here's a listing of the projects. This is just on my development instance that's just running on my laptop. So I just have a couple of example projects there. One thing you might notice is that Irida has a very sort of clean and straightforward interface. So I think it's quite nice, especially in a sort of a outbreak investigation where you're collaborating with bioinformatics folks and also epidemiological folks and maybe some more clinical folks. Then this clean interface helps everyone to sort of get on the same page and access the data in a straightforward way. If you drill down into a single project, then you can see a list of all the samples that are included in the project. And see, I'm not gonna go over every single feature, but again, you can see that some of the more detailed features, like you have access to the line list from here. There's also a really nice automated export into NCBI SRA database if you want to do that. There's integrated quality control. So every pair of FastQ files that you upload will be automatically analyzed using FastQC, and you'll be able to always store and recall quality control information for all samples. And when you have a few samples loaded into your system and you're ready to start doing some analyses, there's a nice clear way of selecting from a limited curated set of high quality analysis pipelines. I think so. The goal with Irida is in sort of a public health setting where you're collecting samples over time, and you want a really consistent, well-documented system for capturing all of your analyses, including provenance information, so that you don't have to think every time you launch an analysis, you don't have to think about every single parameter. But if you ever do choose to tweak a parameter here and there, all of that information will be saved. So here's an example of after an analysis is done, There's a provenance tab that includes every parameter for every tool that is running in the pipeline. So there's, as I mentioned, there's a set of built-in irida pipelines. You can do assembly and annotation using Shovel and Praka. There's a tool to run MASH, both MASH distance and MASH screen against a built-in refseq database. You can do a phylogenomic core SNP phylogenomic tree using Snivel. You can do some salmonella-specific analysis using a tool called Sister and BioHansel. And as we mentioned earlier, Mentalist can do a fast CV and whole genome MLST analysis as well. So that's what you get sort of built in once you just set up your irida instance from the start. Those all come bundled with the system. If you want to go beyond that and look at the other irida pipelines that are available out there, there is a GitHub page under the Public Health Agency of Canada, National Microbiology Lab GitHub organization. There's a irida pipeline plugins page that is a simple README document with a list of the existing irida pipeline plugins. There may be other irida pipeline plugins out there. This is a very sort of federated system where independent parties are able to stand up their own irida instances and develop their own irida pipeline plugins. And people can and are doing that, completely independently of the NML. And I guess one thing that I wanted to just mention here is that if you are out there developing pipeline plugins, or you know of someone who is, then it would be great if everyone who is developing pipeline plugins would list their plugins on this GitHub repo, because it'll help everyone to find the plugins that are out there now. As far as installation goes, installing a new pipeline plugin into irida has to be done by a system administrator. A user of the system cannot independently install a pipeline plugin. And it's a relatively simple process. It consists of first installing any necessary Galaxy tools in the Galaxy server that actually runs the pipeline analysis, and then just copying a single Java jar file into the plugins directory that's configured for your irida server. And then the administrator will have to do a quick restart of the irida system for it to pick up the new plugin and load it into the system. So I'm going to start talking about sort of what the process is and how to get set up developing your pipeline plugins. So there's sort of three components. This is the way that I do things, so this is not exactly the way that you have to do things. Everyone has a slightly different system, and it can be done a bit differently, but I've found setting up these three components in this way to be a productive way of setting things up. So first, I'll just mention that there's some great documentation on this irida.corefacility.ca site for both developers, end users, systems administrators. It's very thorough documentation on the whole system. But that specific link will help you get set up some of the basics of setting up your configuration files in your database. So there are three components to this. You need a Galaxy system. And for that, I would recommend using the Galaxy stable Docker container. So this has become sort of the community favorite developed by Bjorn Grüning. It's a very consistent, simple to use Docker container for setting up Galaxy. I'd also recommend irida needs a MySQL database to store all of its data. And I would recommend also running that inside a Docker container. That makes it really easy to keep it isolated from your, whatever your host system is, your laptop or your workstation. And if you get into a situation where things are getting a bit messy, you've been experimenting and you want a clean slate, that makes it really easy to just wipe the database and start fresh without sort of polluting your host system. The third component is you need the irida source code and an IDE. So the most popular Java IDEs that are out there, IDE is Integrated Development Environment. Either IntelliJ IDEA or Eclipse would be good for that. And if it's not necessary for you to be developing your own Galaxy tools to develop irida plugins, you can use just existing Galaxy tools, but you may find that you need to develop a custom tool here and there. And if you're doing that, then it would be good to pick up this Planimo tool for developing Galaxy tools. So this is sort of an overview of how you can set up your development environment. You have your host system, which is your laptop or your workstation, sort of the actual computer that you're running things on. And that's where you would run your IDE, and you would clone the source code from the irida GitHub repo. And then you have your two containers, one for Docker and one for MySQL. And these circles that are sort of in this boundary between the host and the containers, that just represents, there's a couple of places where you have to map ports. So irida talks to MySQL over a port, and it talks to Galaxy. You want to expose Galaxy over a port as well. So sort of a small implementation detail, but it's just important to get set up correctly. So when you're launching your Docker container, you specify which port is mapped internally inside the container to which port on your host system. And yeah, so this, it's slightly tricky to get this set up the first time, but what I'd recommend doing is just adding a nice little helper function to your .bashrc file if you're on Linux, or your .bash_profile file if you're on macOS. I call mine launch Galaxy, and it contains the Docker command that is necessary to launch the Galaxy container with all of this port mapping set up. And you also need to have a persistent space on your disk where you can store details or store data, persistent data that's going to persist between container launches. So this is exactly the command that I have on my laptop. Oops. And by storing it into your bashrc file, then you don't have to type that all out every time you want to set up the container. You just type launch Galaxy and it goes, it starts up. And if you have a little tweak here or there that you want to add to it, you can just save it in your file and you're ready to go for next time. And same thing for the MySQL container. So the process would be launch Docker, launch MySQL. Once those containers are up, then you can start up your irida system through the IDE, and then you're off and running. Your whole system is up online, and you can start developing. So there are a few constraints on irida pipelines. You cannot build an irida pipeline out of any arbitrary Galaxy workflow. The main constraint right now is that every irida pipeline starts from FASTQ sequence files. I believe that there are some plans to move to a system where you can start from assemblies, but that that's something the irida core developers could talk about in more detail, but I think that's a common question is if every analysis pipeline starts from FASTQ files, then you're going to end up doing a lot of assemblies along the way in your analyses. But what I wanted to explain here is that there are really three ways of getting data into your pipeline plugins. There are your FASTQ files. You can optionally include one FASTA reference file. And then the third place is through Galaxy tool data tables. So in Galaxy, there's a system for persistently storing data that you might want to reuse several times. For example, an index of a human genome or an index of a specific reference genome that you want to be reusing for many analyses. So if you have data stored in Galaxy tool data tables, you can also feed that into irida pipelines. So once your system is set up, you can load up your Galaxy Docker container in your browser. And this is a really nice, nice interactive way of building pipelines. A lot of drag and drop, just selecting tools from your menu and dropping them into the system. And I'm not going to talk too much about the details of building Galaxy pipelines because I have just a few minutes left, so I'll skip ahead. Let's assume you have a Galaxy pipeline. So what else do you need to turn it into an irida pipeline? There's just a couple of additional files that are needed. One is a XML file that defines the parameters and the tools that are necessary for the pipeline. Another one is this messages.en.properties. This is part of the internationalization system for irida, so that you can provide text snippets in multiple languages, but you need at least an English one to get started. So these files can be a bit tricky to set up from scratch, but fortunately there's a nice tool available, this irida-wf-ga to XML, irida workflow Galaxy, going from Galaxy workflow file to the irida XML file. That will automatically generate these two files for you. So that's a really handy tool that's available. The other really handy piece that the NML has provided is an example pipeline plugin. So you can go on GitHub to this link, irida-plugin-example. And what I'd recommend doing is on the right-hand side there's under the plus icon, there's a menu item to import the repository. So what that will do is very similar to forking, but it sort of severs the tie from the original repository. So if you want to start making your own irida plugin, I'd recommend going to that repo, hitting that import menu item, and then you are set up with your own copy of that repo that gets you started with the necessary files to start up your own plugin. So there's just 2 Java classes. That repository contains examples of these 2 Java classes that you'll need. And I don't know how many people write Java or like writing Java, but it's really not a lot of code that is necessary to get this. But there's just a few pieces that need to be put in place. So there are 2 classes. One defines some plugin details. Each plugin needs a unique UUID, and you can use this online tool to generate, or there are a lot of different ways to develop or generate UUIDs. That's one of them. You have to pick a color. You can just Google for a color picker and pop that in. And then you have to define a simple label, unique label for your pipeline plugin. So I have one called species abundance, and I just use the label species abundance. The second component is a piece of code that defines how to load pieces of information from your analysis results into the metadata line list. And that the class has to exist, but that functionality is actually optional. So if you're not interested in that part, you can just sort of drop a stub in there that it exists but doesn't really do anything. But if you do want that functionality, the basic steps are read in one or more output files from your analysis, extract the specific data elements from the that you want from those files, and update the sample metadata to include that new data. And that example plugin has an example of all this code. So it's really a matter of sort of tweaking, starting from that example and tweaking until you get the data that you need. I'm running short on time, so I think I'll skip that. I just had a few sort of general guidelines if you're going to start developing pipeline plugins of your own. I'd recommend including tools that collect QC information in your pipeline. So it's very common in a lot of the existing pipelines for irida. If you run an assembly, then you'll also run Quast to collect some metrics like N50 and to assess the quality of the assembly. You may not feed that information into any downstream tools, but you can just collect it, and then it's there if you need to review it or compile that information later. And similarly, if you're generating a BAM file, you can run BAM stats or something. I'd say I would recommend creating metadata fields sparingly. So each project has one metadata table to work with. And over time, if there are a lot of analyses being done that create a lot of metadata fields, that could become unwieldy. So each time you run an analysis, you can choose whether or not to load any data into the metadata table. But I would say be selective about what you want to load into that table. As with any software, documentation is very important. So that example plugin has a great README file, and I'd recommend just following that example and filling in the details for your plugin. The more details, the better. Potential users will want to know, what does this pipeline do? What tools are necessary for installing the pipeline? So having that all that information up front will be really helpful. And generally speaking, short, simple pipelines are more reliable. If you have a pipeline that's 8 steps deep, there's more and more chance that somewhere along the line, one of those tools may fail. So it's preferable, I think, to have a collection of simple pipelines rather than one master do-everything pipeline. And that's all my slides. So thank you very much.
All right. I'll clap on behalf of everybody. that was excellent. Right, so I've already polled for a few questions, and we'll start off with, are there any plugins for SARS-CoV-2 yet?
I have an experimental version of the Arctic pipeline that I haven't pushed out there. I've only installed it on my development system, and so right now that's the only irida plugin that I know of. There are none listed in the page that I mentioned, the sort of official central list of pipeline plugins. But I think there's tons of potential to get some out there quickly. There are so many pipelines popping up these days, and so finding ones that will fit the constraints of irida and sort of be generally applicable for as many people as possible might be a bit challenging, but yeah, that's sort of where it stands.
Okay. We had another question of, is there a way to bundle tests or test data with a plugin so people can know that it is working locally?
So you can include anything you'd like in your GitHub repo, and you can, I have for some of my more recent pipeline plugins included continuous integration testing, so using that Travis CI integration with GitHub to run some limited integration tests. It doesn't set up a full, it's not a full end-to-end test where you're actually running real data through a full system. It's basically just testing the functionality of the Java classes. So there's no like integrated way to automatically bundle example data with Irida plugins, but certainly you can make, it's a very good idea to make example data available in some reasonable way. I know, for example, the Snivel pipeline in its documentation has a set of example data, the same data that they use in the paper, so that you can download that, load it into your system, and confirm that you get the same results.
Okay. Let's see what else we got on the list. Okay. No, those are all from me. This is one from someone else. I wonder how Irida compares to Nullable by Torsten Seaman, and I guess I'd amend that question with, what is the closest tool out there to Irida?
Well, so Nullable, so one thing I'll say is that, I mean, Torsten has developed an amazing set of tools that Irida uses several of Torsten's tools in the existing pipelines. For example, the assembly annotation pipeline, as I mentioned, is simply shovel and Proca. Yeah, all hail the Torsteverse, right? But Irida is a more flexible system, especially now that this pipeline plugin system is in place. As long as you fit the constraints that I listed, you can build any analysis pipeline that you want and add that into a suite of analysis pipelines. And whereas, yeah, Nullable is sort of a single cohesive predefined set of analyses. Irida lets you mix and match and combine more customized analysis pipelines. And I'm not sure, I mean, there's Galaxy, which allows you to run similar pipelines, but doesn't have quite as cohesive project management picture, and some of the, like the REST API and the NCBI integration is really convenient for Irida.
Yeah, that's good. Is there any other questions from other people? I have one or two I wanted to ask.
Don't want to go too far over time.
Don't want to go too far over time. Where we? Yeah, I think. Yeah, I'll dump mine in the. So there's a lot of conversation going on the Discord in conference room 1. If people have any questions they want to just put in there,
yeah, I'll
go for it.
And
Dan, you'll be wandering around there to catch up with anybody. But yeah, we should press on, and I think we should cross over to Dag. Dag, can you hear me?